Hello, dear student. Here we are again with the continuation to the last lecture, that is drug. In last lecture, we have seen the definition of the drug according to WHO, which of the chemical compounds are said to be drugs. Then we have seen the properties which make drug ideal drug. Then we have seen the different types of drugs like antipyretic, analgesic. We have seen its example. In analgesic, we have seen the most common analgesic drug, sulfanilamide, its uses, its synthesis by most traditional method, which was first discovered by Glumo, and then the modern method of its synthesis. Now, in today's lecture, we are going to see the synthesis of the more advantageous derivatives of the drug sulfanilamide and antimalarial drugs. Okay, so most advanced or the more effective derivative of sulfanilamide is sulfadiazine. Okay, this is the structure of the sulfadiazine. This is sulfa and this is diazo compound, so sulfadiazine. How this molecule is synthesized? In its synthesis, this acetoaniline, N-acetoaniline, in presence of chlorosulfuric acid reacts with the benzene ring. This sulfonyl group reacts with the benzene ring and gets chlorosulfonation at the para position of this N-acetoaniline. Okay, there is para-acetyl sulfanyl chloride is get forms. This para-acetyl sulfanyl chloride on treatment means 2-aminopyrimidine, the hydrogen from this NH and this chlorine from this sulfanyl chloride group gets removed in the form of HCl and we get this derivative. Okay, this N-acetosulfadiazine. Okay, and after treatment of this N-acetosulfadiazine with the aqueous NaOH, this Ac group COCH3 group gets removed in the form of acid and we get this sulfadiazine drug. Okay, this is how sulfadiazine is get synthesized. Then what are the uses of this sulfadiazine? Even we are having sulfanilamide in hand, why do we need to go for the preparation of the sulfadiazine? Okay, so this usage says that why this drug is superior over sulfanilamide. Okay, so this is 8 times more active than sulfanilamide for curing bacterial infections. Okay, 8 times more effective. It is less toxic, as we have seen in sulfanilamide. It is toxic when taken for weeks, so it is less toxic here. Sulfadiazine is less toxic and it is absorbed slowly by intestine and excretes slowly out of the body. Okay. Then third use is it is used in infections like meningococcal, pneumococcal, Friedlander bacillus and influenza bacteria, etc. Okay. So this is how sulfanilamide is gets minimum application than that of its more effective derivative sulfadiazine. Okay. Now next topic is antimalarials. What are antimalarials? These are the compounds which are used to treat the fever of malaria. Okay. In case of malaria, when you get the fever, to treat this fever, antimalarial drugs are used. And the examples of antimalarial drugs are quinine, chloroquine, and plasmodium, etc. This is the structure of the chloroquine. Okay, and when one OH group is attached over here, it is called as hydroxychloroquine. Okay, which is nowadays used in treatment of the coronavirus. Okay, it is derivative of chloroquine, but Most commonly, chloroquine is used in treatment of malarial fever. Okay, how this chloroquine is synthesized? It is this its synthesis is divided into three steps. Out of which, first step is preparation of 4,7 dichloroquinoline. Okay, how this chloroquinoline is formed? Dichloroquinoline is formed. This 7-chloro-4-hydroxyquinoline on treatments with phosphoryl chloride PuCl3 gives us the dichloroquinoline 4,7 dichloroquinoline. One of the Cl from here gets replaces with this OH. What happens? This O bonds attacks over on this phosphorus and then one Cl group gets removed and this negative Cl gets attached over here and the PO2 Cl2 is formed and here we get the chlorination. Then in the second step in the synthesis is preparation of 4-diethylamino 1-methylbutylamine. Okay, this is the bit lengthy process in the synthesis of chloroquine. Okay, or more steps involved in this preparation of this second fragment of the molecule. Okay, so what is going on in this synthesis is acetoacetic ester on treatment with N,N-dimethylamino 2-chloroethane, N,N-diethylamino chloroethane, right? Chloroethane. In presence of sodium, we get this chlorine get removed and this hydrogen from this carbon gets removed in the form of HCl and the bond in between this carbon and this carbon gets formed together to diethylamino ethyl acetoacetic ester. 2 diethylamino 2 diethylamino ethyl acetoacetic ester. Okay, when this diethylamino ethyl acetoacetic ester is treated with HCl, there is decarboxylation. Okay, decarboxylation means removal of CO2. Okay, this removal of CO2. Okay, so here this bond get breaks, this CO2 gets removed, and this C2H6 or C2H5 Cl will form, and we get over here this compound 1-diethylamino 4-pentanone. 1 diethylamino pentanone 4-pentanone 1 2 3 4 4-pentanone. Okay, so if we are going to number the carbons from this position 1 2 3 4, so 4-pentanone. And why 1 diethylamino? Because to the first carbon atom NC2H5 is attached, so to the first carbon atom, so 1 diethylamino and 4-pentanone 1 2 3 4 and 5 carbons are there, so pentanone 4-pentanone. Not if this carbonyl group is present over here, then it is 5-pentanone, pentanone or pentanal. It is aldehyde, so this ON is replaced by AL, pentanal. Okay, if this carbonyl is attached to the last carbon of this chain. Okay, but it is to the fourth carbon, so 4-pentanone, and it is a ketone, so pentanone. Okay, now this compound 1-diethylamino 4-pentanone on reduction in presence of ranitidine and ammonia hydrogen gas, it gives us the removal of this carbonyl group. What happens over here? First, this NH2 gets attached to this carbonyl group and imine gets formed. Okay, now that imine is reduced by the hydrogen and nickel, so that this double bond N having carbon gets converts into single bond NH2 carbon. Okay, and amine. Okay, if this NH2 group is present to the aromatic ring, it is aniline, and when it is present to the aliphatic chain, then it is amine. Okay, so here is the second fragment of the molecules get formed, that is 4-diethylamino 1-methylbutylamine. Okay, as we have said in the heading of this preparation of 4-diethylamino 1-methylbutylamine. So this compound is get formed over here. In the next step, the last step for the synthesis of this chloroquine, is the condensation. What happens over here in this step? The 1,4,7-dichloroquinoline they have synthesized first by treating 4-hydroxy-dichloroquinoline with the 4-hydroxychloroquinoline with the POCl3 to get this compound, and this compound we have synthesized in the last steps beginning with the acetoacetic ester and its treatment with the uh, uh, diethyl amino ethylene chloroethylene chloroethane. Okay, and we get these two compounds or these two fragments we have to attach to each other. This H from this NH2 and this chlorine gets removed to form the bond between this carbon and this nitrogen. Okay, and we with the removal of minus HCl, this hydrogen from here and Cl from here to remove the HCl, and the bond between this nitrogen and this carbon gets formed and we get the molecule chloroquine. Okay, this is how chloroquine is synthesized in 3 steps. Okay, then what are the uses of this anti-malarial agent chloroquine? Okay, it is used in the prevention of malaria, very simple and repetitive thing. It is used to treat malaria caused by various parasites like Plasmodium vivax, Plasmodium malariae, and Plasmodium oval. Okay, etc. etc. These three are the malaria causing parasites, and these parasites will get killed by the this drug chloroquine. So if the patient is deceased by malaria due to these infections, then this infection gets cured by the agent chloroquine. Okay, it prevents what? How does it occur? How does it kills um, these various pathogens is by preventing the transcription, that is protein synthesis, and limits the nucleic material and limits the formation of nucleic material, that is DNA and RNA formation in the reproduction of these insecticides, or not insecticides but the pathogens get uh, get stopped by this drug. So the protein synthesis is not there and the DNA or the RNA material, nucleic material synthesis is not there. So the population of the parasite gets reduced. Okay, and the very usefulness of this drug is that drug can be used during the pregnancy too. Okay, it is very safe to use this drug during pregnancy. Okay, this drug is also used in extraintestinal amoebiasis, rheumatoid arthritis, discoid lupus erythematosus, then lepra reactions, and then the photogenic reactions, and in infection of mononucleosis. Okay, so these are the various types of diseases, and to cure this type of malfunctions in our body, this drug chloroquine is used. Okay, so this is how we have completed the drug part of the chapter 4 of BSc third year. In next lecture, we are going to see pesticides. Thank you.